Mod 4 and Mod 5. You'll probably have like one person for Mod 3 and then 2 for 4 and 2 for 5 is what I would suggest. Everyone will do it one time, but for Mod 3, just make sure it's not the person who had the article that you picked. So someone else would be presenting on like that article's methods that you're going to use for Mod 3. So the point of your pre-lab presentation is basically to give me, Dr. Pai, and Ketki info as to what you're doing, and Dr. Pai, me, and Ketki will tell you, like, is this feasible? Oh, instead of doing this assay that you're suggesting, let's do this one. We already have it. Or like, oh, we can definitely order that. Or like, oh, that's totally something we can't order in time. It's just kind of a way for us to kind of check you before you get started on your experiment. So for the pre-lab presentation that we're doing next week, we're starting at 9:30, not 8. So you don't have to come until 9:30 next week. So what needs to, whoever's presenting the Mod 3 pre-lab next week, you need to give like kind of a sufficient background of the study and the motivation behind it. So why are you interested in like this compound and how it acts on your cell line? You need to tell me the objective of your Mod 3, so what are you trying to accomplish, and a hypothesis. So do you think that increasing concentrations does what cell viability, at what concentration might you think it works best, just whatever your team's hypothesis is, that needs to be in the pre-lab presentation. The biggest part of this is like materials methods, because that's what we're really checking for to make sure that we're all on the same page about what you're going to be doing. So you need to talk about the cell line you're using and why that's like sufficient for this study, why are you using that cell line. The number of cells needed, the treatment groups that you're going to use, how many treatment groups you're going to have. So like you'll probably be using a 96-well plate again if you're doing another kind of like CCK-ish type of assay. So depending on your treatment groups, controls, time point, some people are going to want to see like they tested it 24 hours, 48 hours. I don't know what y'all's papers are yet, but that's just what I've seen in the past. So you need to kind of account for all of these things. You need to have a nice like protocol in place of what you're going to do in the assay, the stats that you're going to run on your results at the end, and tell me what you expect to find, basically. Also, all of this can be found in your handbook. There's a section in your handbook that talks about what you need for the pre-lab presentation, and there's a section in your handbook that is the rubric, how you're going to be graded for the pre-lab presentation. So like I said, only one person presents this next week, 2 for Mod 4, 2 for Mod 5, unless you have 6 people in your group, which I don't know if y'all do, but then 2 people would present next week if you have 6. That's about it. Everyone has 1 grade, so you're only graded on the one that you present, but I would suggest sharing the presentation with your whole team ahead of time, just so everyone's on the same page about what you're about to do for the module. And with that, we can get started. Oh, I also just wanted to reemphasize that Dr. Pai wrote this handbook really, it's really clear and it's very thorough for this class, and I feel like for this module there was a lot more like stress, I guess, than he intended. I just got so many like last minute